Hi and welcome to the next lecture of the course, and we will continue our discussion from the last lecture and start and keep talking about selection in an environment. We had started our first model for selection between two competing genotypes A and B of a bacterial species, and we had noted that if the growth rate of the mutant that has arisen in the population is more than that of the parent or the original genotype, then it is likely that the mutant is going to take over the entire population and eliminate the parent from the population. On the other hand, if the mutant that has arisen is of a lower fitness than the original parent genotype in which the mutant has occurred, in which the mutation has occurred, then the mutation itself is likely to get eliminated from the population. But one thing that we did not account for in our first selection model is the fact that populations could still increase exponentially for an unlimited time and approach infinity. That we have already seen is not a representative of how growth occurs in liquid media for bacterial species or for any species for that matter. And we are going to try and address that issue by building slightly more advanced models of selection which are much more representative of biology and population growth. So, how do we approach this? We approach this by assuming an environment. Let us think of an environment which has a carrying capacity K. This is my environment E, and the carrying capacity of this environment is equal to K. And we have already seen that the carrying capacity of an environment means what is the total number of bacteria or individuals, in this case we are talking bacteria, that can survive in this particular environment. And now let us imagine a scenario where in this environment E, at any given point in time, at any given point of time, we have two distinct genotypes which are coexistent. And let us say that is genotype A, which are represented by the blue bacteria, the growth rate associated with this genotype will be small a. We will just continue the same terminology as what we used in the last lecture. So, A is the growth rate of this particular genotype, and this population, this environment is co-inhabited by another genotype which is genotype B, which are these green shaded circles. This is genotype B represented by these bacteria with these shades. This is genotype B, and the growth rate associated with this particular genotype is B. So, that is the scenario that we are going to be looking at, and we are going to make one more change in the model compared to how we did it last time. What we are going to start with assuming is that the system is operating at a situation at the particular instant at which we started monitoring the system. The number of individuals in the environment taken A and B together is equal to K. The system is operating at its carrying capacity. That means number of A individuals Na plus Nb is equal to K. And selection basically takes place. The basic tenet of selection is based on competition, that there is resource limitation and individuals are vying with themselves for the limited amount of resources which are available in the environment, and those which are better adapted to the environment take more advantage and grow further, their numbers increase further in the environment, and those individuals which are less suited to survive in that environment get left behind and are eventually eliminated. And what we are interested in monitoring is that in a system like this where NA and plus NB is equal to K, that means the system is already operating at its carrying capacity. How do NA and NB change with time while the total population in the environment is maintained at K? That's the question that we are trying to address via this model. And to do that, in this framework, we are going to not talk about numbers of bacteria, but we are going to define two variables, which is Na by K plus Nb by K equal to 1. So, I have just taken K from here to the other side and arrived at this relationship. So, let me just rewrite that equation again, and we have this relationship that in this 2 bacterial system, the number of individuals when added up for the both types equals K, and I can rewrite that equation as Na by K plus Nb by K equals 1. And I am going to write this as Xa plus Xb equals 1, where Xa represents fraction of population which belongs to genotype A. Similarly, Xp represents fraction of population which belongs to genotype B. This is a very simple but a very important statement. Imagine that we are operating at a scenario where Na is equal to K and Nb is equal to 0, which has to be true because we know that at any instant of time Na plus Nb is equal to K for the system. In such a situation, Xa will be K upon K. Remember, Xa is being defined as Na divided by K. So, Xa is equal to K by K, which is 1, and Xb will be 0 by K, which is 0, right. So, the fraction of population which belongs to genotype A in such a scenario is equal to 1, and the fraction of population which belongs to genotype B in such a scenario is equal to 0. If Na, let us take one more simple example, if Na was K by 2 and Nb was K by 2, then you simply have Xa equals half and Xb equals half as well, right. But at all points in time, no matter what are the individual numbers that these 2 species are operating at, we have the fraction of individuals, fraction of the population which belongs to type A plus fraction of the population which belongs to type B. When these 2 are added together, you get a sum constant equals to 1, and which is because of the fact that the total population size is equal to K at all instants of time. If that is true and we start with this relationship, we have Xa plus Xb equal to 1. I want to understand how does Xa and Xb change with time, because that is the basic quantity that I am interested in, that should I have A and B genotypes coexist in a similar environment, how do these fractions Xa and Xb change as time progresses forward. That is the basic idea that I want to understand. When selection acts, how do these frequencies change, or how do these fractions change. So, the rate of change of Xa and Xb with time is the quantity that I am interested in. So, I am going to differentiate this simple equation that I have with respect to time. So, I get d by dt of x a plus x b, and I differentiate the right hand side with respect to time as well, d by dt of 1. Now, derivative of a constant is just 0. So, I get a 0 on the right hand side, and the left hand side gives me dxa by dt plus dxb by dt equal to 0. This again is a very important result, because what this tells me is that rate of change of xa with time plus rate of change of xb with time is equal to 0. In other words, I can write this as dxa by dt equals minus dxb by dt. This is an important result here. Because the total number of individuals in a population is conserved to k and the total number of individuals does not change with time, at any given point in time we have k individuals, some of them belonging to type A, some of them belonging to type B. Now, we are interested in understanding how do these numbers belonging to type A and type B change with time. But because the population size total is constrained to be equal to k, dxa by dt, which is how do numbers of type A change with time, is equal to and opposite in sign to dx by dt. So, if you look back, if you look at the equation again, what this means is that if fraction of type A goes up by 0.1, okay, let us imagine that xa goes up from 0.5 to 0.6. What all of this analysis means that if this ratio changes by 0.1, this has to change by minus 0.1, so as to ensure that the total number of individuals remains equal to k. If during selection number of 10 individuals belonging to genotype A are eliminated, that means 10 individuals belonging to genotype B must have arisen to make sure that the total population size is held constant at k. Hence, the rate of change of the two genotypes is related via this relationship. So, where does this take us? We have this relationship between the rate of changes of the frequencies of the two genotypes, and we also have the relationship that xa plus xb is equal to 1. We are going to take these forward and try and develop our model for selection. So, now I am interested in dxa by dt equals xa times a minus phi, and dxb by dt equals xb b minus phi. Phi is an unknown that I am not sure about at this point what is it, but I want to find that out, and I want to introduce phi to impose some sort of resource limitation on the model, because I know I am working for a model where the population size cannot exceed k even when both the genotypes are added together. In fact, when all the individuals are counted belonging to both genotypes, the population size in this model has to be equal to k. But I am not sure what is that variable phi that I should introduce in my model so as to ensure that the population size does not exceed k. In the last lecture, we had developed the most simplest selection model. We had put phi equal to 0, and that would give me dxa by dt equal to a times xa and dxb by dt equal to b times xb. That model had meant that the numbers of individuals belonging to each species could go to infinity. And that is the particular scenario, that is exactly the scenario that we want to avoid here and constrain the population size to not go beyond k. And for that reason, we are introducing this variable phi, which we do not know at this point what is it. So how do we find phi? Remember this result that we just obtained, that dxa by dt plus dxb by dt is equal to 0, and we are going to make use of this. To make use of this, in the model that we have, we are going to add the two. We know dxa by dt, we already have an expression for that, and to that we are going to add dxb by dt. And we know from the analysis that we just completed that these two differential derivatives when added together must be equal to 0. So this is equal to 0, this we know from the previous slide, but what we have done here is derived expressions for these derivatives, and this gives me xa times a minus phi plus xb times b minus phi. I solve these together, and let me do that on the next slide. I get 0 equals a times xa minus xa times phi plus b times xb minus xb times phi. I am just going to do some rearranging. I will take the phi terms over to the left hand side, and that gives me xa times phi plus xb times phi equals axa plus bxb. And this can be rewritten as I take phi common and I write xa plus xb equals axa plus bxb. But again, let's go back to the two results that we derived 2 slides back. I know that xa plus xb is equal to 1, and I am going to use that result here because this is xa plus xb, and I have already shown that this sum is equal to 1, and hence phi is equal to a times xa plus b times xb. So using the model that we started with an unknown phi, which we did not know how to explain, but having imposed the constraint on the model that the population size of both genotypes put together has to be equal to k at all times, I have obtained an expression for phi. Now what does this phi mean? This phi has a very important biological meaning associated with it. This is the expression that I have for phi. Just look at this expression carefully. What is A? A is the growth rate associated with genotype A, and B is the growth rate associated with genotype B. XA is a fraction of population which belongs to genotype A. XB is the fraction of population belonging to genotype B. So what this tells me is that growth rate of A multiplied with fraction of population belonging to genotype A plus growth rate of B times fraction of population belonging to genotype B. This means that this is just equal to the mean fitness of the culture. What this means is that this is the mean growth rate of the culture when I have my population as XA fraction belonging to type A and XB fraction belonging to type B. Let me illustrate that with a couple of examples. Suppose XA is equal to 1 and XB is equal to 0. What does that mean? That means that all individuals in the population are of type A. What that implies is if all individuals are of type A, the average growth rate should be equal to just A because there is no individual of type B. And what does phi give me? That gives me A times 1 plus B times 0, which is A. On the other hand, if XA is equal to half and XB is equal to half, because their sum has to be equal to 1, we have already derived that. Then what is the mean growth rate that you associate with this culture, which has half the cells growing at rate A, half the cells growing at rate B? And intuition tells us that growth rate should be the average of the two growth rates A and B, and if we plug the values in this relationship, we get A into half plus B into half, which is just equal to A plus B by 2. Again, this goes to illustrate that phi has a very real biological meaning, which is the mean growth rate of the culture when the culture is existing at frequencies or fractions xa and xp. So let's go back to our equation and use this meaning of phi in the selection model that we had developed. If we do that, we get dxa by dt equals xa times a minus phi. Now phi we have already derived is xa times a minus a times xa plus b times xb, which can be rewritten as xa a minus axa plus b. I am going to write xb as 1 minus xa, because I know xa plus xb is equal to 1, hence this can be written like this, and I get this equation as xa. From these two terms I can take a common, I get 1 minus xa. This should be a negative here, because when I open the bracket there is a negative sign outside. I take a common and I am left with a times 1 minus xa minus b into 1 minus xa. And now from these two terms I take this common factor 1 minus xa out, and I get xa times 1 minus xa into a minus b. That is my first model for selection, which tells me how does frequency of A change with time when it is competing with another genotype B, which is growing at growth rate B. So we will spend the next few minutes analyzing the dynamical properties associated with this particular equation, using some of the concepts from 2 lectures back where we were talking about stability of these systems. So how do we do that again? We have our differential equation dxa by dt equals xa times 1 minus xa times a minus b. Now remember from how we did this in the previous class, the first thing that we need to do is plot the curve between x and f of x, where f of x is just the expression that dxa by dt is equal to. But the nature of this graph that we have for this situation is actually going to depend on the parameters associated with this model, which are the individual growth rates of the 2 species A and B. If A is greater than B, then I am going to have one particular shape of the curve, but if B is greater than A, I am going to have a different shape associated with the curve. So I need to analyze this system differently depending on whether A is less than B or A is more than B. So what we are going to do is, for the case A greater than B, the steady states of this system are whenever we to calculate the steady states, we put dxa by dt equal to 0, and that tells me that this is true when I put this entire expression equal to 0. This is true whenever xa is equal to 0 or xa is equal to 1. These are my two steady states associated with the system, which ensure that either this quantity is 0 or this quantity within the brackets is equal to 0. A minus B does not come into picture when I am computing the steady states of the system. Now, we discussed that the plots are going to look different when I am trying to plot fx, and that will depend on whether A is bigger than B or B is bigger than A. If A is bigger than B, that means this quantity here is a positive quantity. If B is bigger than A, that means this factor is a negative factor. So, and my two steady states are 0 and 1. If A is bigger than B, the plot looks something like this. This is for A bigger than B, and if B is bigger than A, the plot changes its nature and it looks like this. So, the green curve is for when B is bigger than A, and the red curve is for the case where A is bigger than B. So, I leave this as an exercise, and we will begin the next lecture by solving the stability of this system using these two scenarios and see what that tells us about the biology. But before we do that, I would like you to try these two scenarios yourselves and comment on the stability of the two steady states we have in either cases when A is greater than B. What can you say about the stability of the two steady states xa equals 0 and 1, and what can you say about the stability of the same steady states when B is bigger than A? And more importantly, what do they tell you about the biology of the problem that we are talking about, two competing genotypes A and B in an environment where population size is constrained to be equal to k at all points. So, we will pick that up when we start the next lecture. Thank you.